Hello, my name is Paul Atkins, and I'm very pleased to share with you my thesis work from Dan Voitas's lab at the University of Minnesota. I studied gene targeting in plants. In particular, I developed a method to analyze gene targeting outcomes and mechanisms in somatic plant tissue using nanopore sequencing. The high-throughput molecular analysis of gene targeting events is made technically challenging by the residual presence of donor molecules. Donor molecules restrict primer placement, resulting in large amplicons that cannot be readily analyzed using standard NGS pipelines or qPCR-based approaches such as digital droplet PCR. In somatic plant tissue, removal of excess donor is time and resource intensive, often requiring plant regeneration and weeks or months of effort. This imposes a severe bottleneck on gene targeting experiments, often limiting throughput to at most dozens of gene targeting outcomes for analysis at the nucleotide level. Here, we utilized Oxford nanopore amplicon sequencing to bypass limitations imposed by donor molecules and to dissect gene targeting outcomes at 3 loci in Nicotiana-Benthamiana leads. Using this, we captured tens of thousands of gene targeting events and thousands of conversion tracks, all without selection or prolonged propagation. Nanopore amplicon sequencing allowed us to observe the low frequency of somatic plant gene targeting using donors with 500 base pair homology arms, Cas9, and guide RNA, and the ability of Gemini viral replicons to enhance gene targeting frequencies. Using donor molecules with imperfect homology, in this case regularly interspersed SNPs, for monitoring conversion tracks, we show both the size and distribution of thousands of conversion tracks in leaf tissue 5 days after reagent delivery. These data demonstrate the usefulness of nanopore amplicon sequencing for analyzing plant gene targeting events and provide a mechanistic basis for future plant gene targeting optimization. I hope to talk with you about this more at my poster. Thank you.
Gene therapies could probably help us to live much longer and healthier lives in a not so distant future. For this, we do not need more genetic data. We have already gigantic data. For this, we probably do not need so much new artificial intelligence. We have already thousands of apps, thousands of computer scientists, and many usable software. And for this, we do not need to convince citizens that science should help us to live longer and better lives. Citizens are already in favor of the sharing data for health research. But we need to share data better than now in a more altruist way. We need to have a kind of a global project of editing genomes for common good and for common health. Gene therapies for healthy longevity are very probably a big part of our future. If we can work together with altruist goals, then we can make this a closer future for all. Thank you.
Hello, my name is Nathan Feltman. I'm a graduate student in the Szymanski Lab at the University of Minnesota. Invasive insect pests are a growing concern for human health and food production. Current insect biocontrol methods rely heavily on insecticides, which can harm beneficial insects and lead to the evolution of pesticide resistance. In the Szymanski Lab, we have developed a technology called Engineered Genetic Incompatibility, or EGI, which can be used as a species-specific biocontrol approach. EGI uses a CRISPR activation mechanism to induce lethal overexpression of a tightly regulated gene, but only in organisms which are not homozygous for a resistance mutation. Releasing male EGI insects into the wild should decrease the pest insect population of the next generation by inducing lethal overexpression in EGI wild-type hybrid offspring. A limitation of EGI is its reliance on conserved promoter sequences to drive hybrid lethality. We've developed the huge analysis pipeline to screen target promoters of wild pest populations for genetic variations, such as single nucleotide polymorphisms. By using our huge pipeline on thousands of wild spotted-winged Drosophila, we can rationally design our EGI components to maximize their effectiveness at reducing the number of spotted-winged Drosophila in the field.
Hi, I'm Evan Kleinbel, a graduate student in the Moriarty Lab at the University of Minnesota. My project is about the development and testing of a universal base editor reporter platform. Base editor proteins are novel fusion proteins engineered based on the CRISPR-Cas9 system that converts single nucleotides into another nucleotide. This offers a new method of gene therapy. However, in vivo gene editing presents new risks and challenges, such as off-target mutations, efficacy of the vehicle to deliver these tools, and the DNA repair mechanism inside these cells. Without addressing these problems, it would be impossible to develop safe and effective in vivo gene editing. We constructed a universal base editor reporter called Uber. This allows us to develop and test novel base editors in vivo. If you'd like to know more, come visit my poster.
Hello everyone, my name is Hitesh Kumar. I am working as a postdoctoral associate in Funk's lab at the University of Minnesota. And in this lab, my goal is to achieve high throughput precise genome editing in plants. So we try to improve the frequency and precision of targeted insertion in this study by understanding the role of chemical modification on donor DNA, and our results suggested that 3' modification of the donor significantly improves the targeted insertion frequency. And in this targeted insertion, we found no orientational preference of targeted insertion, and further we improved the precision and control the orientation of the targeted insertion up to 80% by using a rationally designed donor. And further we try to leverage this technology to efficiently tag endogenous genes in plants and their detection using an antibody-free approach. So together my studies suggest a great potential to combine precise genome modifications with other high throughput approach for basic and translational research. So therefore I look forward to see you for more detail at my poster.
Hello. Invasive Burmese pythons have been linked to severe mammal declines in southern Florida, up to 99% in some species. Control of these large constrictors is hindered by low detections and capture rates. Furthermore, there is strong evidence the species is expanding its range to the north. The severe impact of pythons on the greater Everglades ecosystem merits an investigation into alternative management methods. Genetic biocontrol may benefit landscape management by not being reliant on human detection of an organism and enabling species-specific applications. Some prominent examples include RNA-based toxicants and CRISPR-modulated artificial gene drives that can target genes that skew sex ratios that could potentially promote population collapse. Research in this complex field requires a commitment to open and responsible science that adheres to thorough risk assessments and following proper permitting and regulations. Moreover, we recognize that it is critical to establish a stakeholder engagement framework to help seek feedback on the research efforts. Douglas Menke's group at the University of Georgia reported the successful generation of crispins in Anolis agrae via CRISPR-Cas ribonucleoprotein delivery to unfertilized oocytes. This method could facilitate transgenesis in non-model organisms like Burmese pythons. One preliminary target is tyrosinase, a gene whose mutations are linked to albinism. This gene is an ideal proof-of-concept target with a visible phenotype that can inform of the efficiency of transgenesis and mutagenesis. Next steps will focus on the empirical validation of the targeted sites and the assessment of off-target effects. Of note, no genetic material or live animals will be released during this preliminary investigation. Thank you.
Hi, my name is Gabrielle and I'm a graduate student in Brandon Moriarty's lab. My project aims to develop a novel NK cell-based immunotherapy to more effectively target osteosarcoma. Osteosarcoma is the most common primary bone malignancy and most often affects children and teens in their second decade of life. Although our understanding of osteosarcoma tumor biology has improved, treatment options and patient outcomes have remained stagnant for over 4 decades. Immunotherapies have gained traction as a cancer treatment, but have been unsuccessful in the treatment of osteosarcoma patients. Natural killer, or NK cells, present a less explored alternative to the more commonly used T cells. With multiple modalities of cancer cell killing and high safety profiles, there is an increased interest in using NK cells for immunotherapies. Unfortunately, many cancers are able to inhibit or even evade NK cell effector functions. We hypothesize that NK cells that have been genetically engineered can be used to successfully treat osteosarcoma. We propose to knock out genes that inhibit NK cell functions and implement CARs that optimally activate NK cell antigen-specific killing in order to unleash NK cell effector functions. Come to my poster to learn more.
Hello, my name is Adam Sekla, and I'm a graduate student in Dr. Mike Smansky's lab. The discovery of antibiotics revolutionized healthcare, where once deadly diseases such as staph or typhoid became trivial. On a day-to-day basis, we take this for granted. However, decades of overuse led to a rise in antibiotic resistance. In the US alone, antimicrobial resistant bacteria cause almost 3 million infections and over 35,000 deaths every year. In the meantime, the discovery of new antibiotics has been steadily dropping off. Even though a whole host of antimicrobial proteins have been identified, they've seen limited use clinically due to their low bioavailability. To address this issue, we developed influenza A virus as a transient, non-integrating gene delivery platform to generate antibiotics directly at the site of infection. Influenza A virus has a genome consisting of 8 negative strand RNA segments and does not go through a DNA intermediate. We redesigned the segment encoding for hemagglutinin to instead encode for lysostaphin, a staph-specific antimicrobial protein. Cells transduced with these particles produce lysostaphin, but because they do not encode hemagglutinin, they are unable to produce new infectious particles. We have demonstrated that using the system, we are able to produce lysostaphin both in vitro and in vivo, and can completely inhibit a cell culture contamination of Staph aureus with as little as 1 viral particle for every 200 cells. Not only that, but this system has been effective against antimicrobial resistant strains of Staph aureus. Our novel platform expands the arsenal in the fight against antimicrobial resistance. If you have any questions about our platform or its use, please come visit my poster.
Hi everyone, my name is Pat Claudio and I'm a graduate student in the Moriarty lab at the University of Minnesota. My project centers around the genetic correction of a rare form of severe combined immunodeficiency, or SCID, in a native population that shares Southern Athabascan languages. This form of SCID arises from a mutation that creates a premature stop codon in the Artemis protein, and in order to correct this mutation, we are using 2 different systems. The first one is the CRISPR-Cas9 system to create a double-stranded break that will induce homologous recombination, where we can revert this mutation back to the wild-type sequence by using a donor template. The second one is the adenine base editor, or ABE, system that creates single base changes from adenines to guanines, which can create a conservative substitution to a tryptophan amino acid that we think should rescue the Artemis protein and function. If you'd like to hear more about my project, come to my poster on Wednesday, July 28th.
Hello, my name is Trevor Weiss, and at this conference, I'm going to be presenting a poster titled Differential CRISPR-Cas9 Genome Editing Influenced by Epigenetic Factors. And so I'm really interested in trying to understand the features that influence CRISPR-Cas9 genome editing. And to approach this question, you can think about it two different types of features, genetic and epigenetic. And so what I did is I wanted to fix the feature of sequence variables so that I could start asking what non-sequence features might be influencing CRISPR-Cas9. So if that sounds like something that is interesting to you, please stop by my poster.
Hi, I'm Jordan Welker, a postdoc in Didier Stainier's lab at the Max Planck Institute for Heart and Lung Research. I'm interested in understanding the mechanism that underlies transcriptional adaptation, and I'm using a transgenic approach in the model C. elegans to try and parse how this functions. So transcriptional adaptation is one of the mechanisms by which an organism maintains genetic robustness. Basically, for mutant alleles, these mutant alleles are generally degraded, and following degradation, you have transcriptional adaptation is describing the process where these degraded fragments are then used to alter the regulation of similar genes. So in this case, we have a premature stop codon containing allele for the gene actin-5, and following degradation of this transcript, we find that the adapting gene actin-3 is upregulated. And previously, we showed that using a transgenic reporter line for actin-3, so this is the promoter of actin-3 driving RFP. It's normally expressed in the pharynx of C. elegans. This line, in the presence of the actin-5 PTC allele, actually gains an expansion of where the RFP is expressed. So you can visualize when transcriptional adaptation is happening using these lines. And so what I've done here is performed a series of truncations and modifications of this transgenic line to try and parse which sequences in this promoter region are necessary and which sequences in this promoter region are sufficient for inducing a TA response. And what I found is that there's about 25 base pairs that are both necessary and sufficient for transcriptional adaptation to act on this reporter construct. And these 25 base pairs can then be used to try and identify motifs to look for what is important for transcriptional adaptation to function on other genes. So if you'd like to learn more, please visit my poster and come talk to me. Thank you.